Thank you for coming to my poster presentation about how persistent alterations in astrocyte-related metabolites in the thalamus is a specific fingerprint associated with the development of post-traumatic epilepsy in mice. Post-traumatic epilepsy is a severe neurological consequence of traumatic brain injury with a 30-year cumulative incidence up to 17%. The latency between TBI and the onset of seizures offers a therapeutic window for the investigation of neuropathological mechanisms and for intervention. We have recently characterized a mouse model associated with 50% PTE incidence by 5 months and a seizure-free period of around 1.5 months post-TBI. In this study, we used our model to investigate brain metabolites changes underpinning PTE development by in vivo longitudinal protomagnetic resonance spectroscopy, or 1-proton MRS, with the aim to identify mechanisms leading to PTE. To do so, we used CD1 mice. We had 8 naive mice and 15 mice that underwent severe TBI by controlled cortical impact with an injury depth of 2 millimeters. TBI mice were also implanted with cortical screw electrodes as shown in the image. Mice were longitudinally followed and assessed for behavior at 1 week and 2 months and underwent 1-proton MRS at 3 weeks and 3 months post-TBI. At 5 months, animals were EEG recorded for PTE versus no seizure categorization based on the presence or absence of spontaneous recurrent seizures. We found that PTE incidence was 46% with an average of 0.3 seizures per day. We also found that PTE mice showed lower recovery of sensory motor deficits than no seizure mice, in line with our previously published data. We then performed 1-proton MRS in the thalamus, a key area involved in epileptogenesis. We found that the levels of the metabolites glutamate plus glutamine, regulated by astrocytes, and myoinositol, an index of astrocyte activation, are persistently increased only in PTE mice. We then went on to explore what happens in the ipsilateral cortex, and we found evidence of higher GFAP reactivity, so increasing reactive astroglial cells, both by Western blotting and immunohistochemistry, only in PTE mice. Overall, our data suggest that alterations in metabolites associated to astrocytes and astrocyte reactivity within the thalamocortical circuit emerge as a neuropathological mechanism leading from TBI to PTE. With this, I want to thank my lab and all the people involved in this study, and thank you for the attention.